Hi to all. My name is Wojciech Sokołowski and I am a PhD student. Today I want to tell you about my PhD research and the main purpose of my study. Also of the rule of expected results in real life. Thank you for your interest and presence. Let's start to listen about natural potential of endophytic bacteria in plant growth promotion and crop protection. At first, the definition. Endophytic bacteria, or wider endophytic microorganisms, can live at least part of their life inside plant tissues, and it is important without causing any disease symptoms. So interactions between endophyte and their plant host can be only neutral or protectionist, beneficial. Why beneficial? Because of bacteria strain-specific biochemical properties. By what bacteria uh, can contribute to promote plant growth and development, and also phytoremediation or protection plants against pathogens directly by competition with phytopathogens or by triggering induced systemic resistance. I said endophytic bacteria could promote plant growth and development. What does it mean? Bacteria can possess the ability to enhance of their plant host growth by nutrients uptaking facilitation, fixation of the nitrogen from atmosphere, and enzymatic solubilise the phosphorus from the inorganic compounds. These two the most important elements for plants. Next, bacteria can produce phytohormones and substances involved in phytohormones level modulation in host tissues, such as deaminase of aminocyclopropan carboxylase, ACC, which is the plant stress hormone precursor. Bacteria can also produce signaling molecules, which triggering mentioned previously induced systemic resistance, the plant's natural defense system against phytopathogens and herbivores. Summing up this knowledge, we know that some bacterial strains in nature have an ability to enhancing plant growth, to protect them against biotic and abiotic stressors, and more. Bacteria can be also used to contaminated soil remediation, and there are the reasons why the scientists from around the world study the endophytic bacteria and their ecological potential. The strains with confirmed beneficial features can be used in agriculture by application of so-called biopesticides, or biological pesticides, which contain live strains of bacteria or their metabolites instead of synthetic chemical compounds, which makes them safe for the environment. In which way can we study endophytes? The general way to study the potential of endophytic bacteria to plant growth promotion and plant protection is included in these three steps presented in the diagram. The first step is acquisition and classification of strains, of endophytic strains. Next, their ability to release various mechanisms of plant growth promoting and plant protecting is assessed, and at last, discovered beneficial features must be confirmed in vivo by plant tests. What about the strain acquisition? We are studying endophytes, so the source of these microorganisms are only the internal tissues of the plant, and the richest source of them are the roots, but also stalks and leaves. In the next way, the bacterial isolates are classified to genus and species. The housekeeping genes and the 16S rRNA gene are generally used to classify. In the second step, previously classified strains are undergoing many in vitro tests which indicate the beneficial features of tested bacteria. What types of tests are usually carried out? Usually, scientists assess the ability to decomposition of inorganic and insoluble compounds, the production of siderophores, which are low molecular compounds facilitating the transportation of iron ions and other metals. Next, the use of alternative sources of carbon and energy, such polycyclic carbohydrates, or even pesticides, chemical pesticides too, which indicates the ability of the strains to degrade these compounds. Also, the production of various enzymes, phytohormones, and antimicrobial substances are tested. Figure number 3 is the photo of the result of simple test of mentioned siderophore production. This discoloration of the medium around the bacterial colony is positive results and indicate that this strain is able to produce molecules which can uptaking iron from the medium. At last, the selected beneficial bacteria are tested in vivo, where the plants are inoculated by tested strains, and beneficial features are tested in quasi-natural conditions, in greenhouse, or in natural conditions, in the field and an area of open land. And the method in which the plant experiments, the plant tests are carried out depend on the previous results, because in this step we must to confirm or decline our in vitro results. For example, when the ability to solubilize phosphorus by tested bacteria were in vitro confirmed, in the plant tests, the insoluble compounds on phosphorus will be used as the sole sources of this element of phosphorus in artificial compost substrate used instead soil, and in this substrate inoculated plants will be cultivated. Then, after cultivation, the growth of both group of plants, the dry mass, for example, of both group of plants are compared, and on this basis the ability of the strain of this strain or strains to dissolve insoluble phosphorus compounds in natural conditions or in quasi-natural, yes, in greenhouse, in pots, can be confirmed or declined. Well, validated in this way, bacteria can be propagated and used in agriculture as biopesticides, biostimulators, or plant vaccine. And the phenomenon of these substances, of biopesticides, is in fact imitation of the natural processes which is still realized in all plants, because probably all plants are correlated with endophytes, and in this way the biopesticides are the real alternative for chemical pesticides, and it was even clearly emphasized in the recently announced European strategy for the environment protection, called the European Green Deal. Now the market of biopesticides is still small, but is constantly growing, and the main difficulty in the development of biopesticides are the high costs of registration of these substances, and the international requirements of their effectiveness at the level of chemical pesticides. But I believe that the development of this field of biology, of this branch of agriculture, will naturally lead to a significant reduction of the use of agrochemicals in agriculture. Thank you very much.